Hello, students, and welcome to my video on cells for Unit 6 assessment. Just like all my videos, this is not for profit. It's just meant to help students. It uses many images from Khan Academy, which is available for free at khanacademy.org. I also encourage you to check out the CK12 textbook that we use in class. The main parts of this video will go over prokaryotic and eukaryotic cells, cellular organelles, and how they contribute to emergent properties of jobs within the cells, and then we'll finish up with endosymbiosis. I hope you enjoyed this unit. There are a lot of fun labs and fun activities. And just remember that cells are the basic units of life. Let's jump in. Make sure to stop the video often, ask yourself questions, and construct your own explanations. Do not just memorize what I say. That is not helpful to you, and you'll forget it in a day. But if you build your own models and ask your own questions and get involved in it, and think about, well, what happens if the mitochondria doesn't work? What happens if the lysosome doesn't work? Then you might get really interested and you can really learn the material, and it could lead to an awesome job when you get older, something that ChatGPT can't do yet that you could research and figure out. Let's check out the study guide. So we started the unit by looking at the cell theory, and there's a fun TED Ed video on that. You don't need to memorize the cell theory, but you just need to know that cells are the smallest things that can be alive. We then compared and contrast prokaryotic and eukaryotic cells. I encourage you to use a Venn diagram for that. Same thing for plant and animal cells. We did a lab on why are cells small. We looked at and used cellsalive.com to go over the functions of various organelles in the cell. Just like in biochemistry, we say that the structure of an organelle leads to its function. We went over a couple of jobs that cells have. And so the first one being energy capture and storage and use. The second big job being making proteins. And the third big job making new cells. We then said that we used the Learn Genetics Utah website to check out how cells can combine together to make tissues. Tissues can make organs. Organs can make organ systems, and they can make an organism. And last but not least, we did the endosymbiotic theory. All right, the cell theory. Once again, no need to memorize this, but I thought it was a fun TED Ed video to show you, the science behind it and some of the work that went into coming up with learning about cells, etc. But the biggest thing to take away from this is that the cell is the smallest unit of life. If you go below a cell, it's not alive on its own. Cells are composed of organelles, and those organelles give it the emergent property of life. Those organelles are made up of proteins, lipids, and carbohydrates, nucleic acids that we learned about earlier in this unit. They're made up of those biological macromolecules. Here's a nice little picture showing kind of the scale of what we've been going through. In many biology courses, you start small here at the atom and you go all the way through to ecology. We didn't do that. We're following this curriculum from California, Next Generation Science Centers. We actually started big and then we kind of went small, and then we went back to big with evolution on a community scale. Then we went into DNA, so we went really small. So we've jumped around a bit, but now by the end of the year, it should kind of all make sense to you. And so we have atoms, right? And they combine together to make molecules. A molecule that has different elements in it is called a compound. And so here is water, that those form together to make macromolecules, carbs, lipids, proteins, nucleic acids, and they can make organelles. Right? And that's those organelles in the cells and tissues is where we'll be at in this video. All right, let's jump in. So the cell is the smallest unit of life. The cells, when they're combined together, they can make tissue, like a muscle tissue, which can make an organ, like the bladder, and then that can be part of the organ system. So here, an excretory system. Those organ systems combine together to make us an organism. All right, why are cells small? There was our fun lab up here. So I made some agar and I dyed it with an indicator. And you got to cut it up and you put it inside of an acid. This would be vinegar. And it started to diffuse and change color. So why do cells have to be small? Like, look at this scale. This is a logarithmic scale. So this is by powers of 10, right? Getting much, much, much smaller until we get to cell size. Here it is. The biggest one to take away from this is that cells have to be small in order to maximize surface area to volume ratio. The surface area is the outside of the cell, and the volume is how much area or how much space it takes up. Right. So the surface area is the area on the outside. The volume is how much space it takes up. Cells have to be able to get in oxygen and food and get rid of carbon dioxide and waste through the process of diffusion. Right? And so they have to be able to get oxygen into their mitochondria, and they have to be able to get rid of the CO2 in an efficient manner. If the CO2 builds up, it becomes acidic. If the oxygen can't reach the mitochondria, then they would die. As an object gets larger, so this is a cube. So we go from 1 by 1 by 1. If we go to 2 by 2 by 2, it's now going to have it'll have a larger surface area, but the volume increases at a quicker rate. It's because volume is to the third power, whereas surface area is to the second power. So the surface area to volume ratio of this 2 by 2 by 2 cube is smaller than the surface area to volume ratio of this 1 by 1 by 1 cube. This one is 6 to 1. That's a ratio, right? Or you could say 6 over 1. Either one's fine. This would be 24 over 8, so 3 to 1. We then did a rectangle cell, right? And it was kind of in between the two, but it wasn't as fast as a small cube. So cells have to be small because they have to be able to diffuse oxygen in and get rid of CO2. All right? They cannot be like, I can't be one cell. I want to be able to get the oxygen all the way in to me that I need. All right. So diffusion is fast and efficient over short distances. It's slow over longer ones. Cell size is also limited by how quickly DNA can make proteins. Cells need lots of proteins to perform critical functions. So if you had a gigantic cell and you just have one nucleus of DNA, it wouldn't be able to get the proteins everywhere quick enough. And then surface area to volume ratio is what we just talked about. Let's check out cell size comparisons. So bacteria are the smallest. They're around first. These are prokaryotes. And then animal cells and plant cells will be larger. You can see that this bacterial cell will be about 1/10 the size of an animal cell. Here is a organizer. And this organizer is actually from the first year I taught in 2004. So here, cells can either be prokaryotic or eukaryotic. Prokaryotes are bacteria. They have no nucleus or pre-nucleus, no organelles. Eukaryotes have a nucleus and they have organelles. All right, so see if eukaryotes encompass plant and animal cells. See if you can compare and contrast these. Try and make a Venn diagram on a piece of paper or a T-chart and see what you can come up with here. All right, maybe you got something like this. Prokaryotic cells have no nucleus. Here was the picture of it. This one right here, it has no nucleus. It has a region of DNA called a nucleoid, but it has no nucleus. It doesn't have anything that encapsulates this DNA. All right, it's free floating and it's circular. They have no membrane-bound organelles. Right, the membrane that they have is on their outside. That membrane can be encapsulated by a cell wall. They are very small. They are older. Eukaryotic cells evolved from prokaryotic cells, and they're only bacteria. There is a huge diversity of bacteria. There are archaebacteria and eubacteria. Don't think that there's just one type of bacteria. Eukaryotic cells have a nucleus, so was infolding of the membrane or invagination of the membrane to form a membrane around the DNA. That enabled that DNA to become linear and linear chromosomes for faster DNA copying. Eukaryotic cells have many organelles. These organelles are membrane bound. They're larger than prokaryotes. They're younger. They evolved from prokaryotes, and they include plant and animal cells. All right, try and compare and contrast plant and animal cells. What do you remember from in class? Make a Venn diagram or a T-chart. All right, hopefully you got something along the lines of that plant cells, they're going to have cell walls. They're more box-shaped. Animal cells do not. Plant cells are going to have chloroplasts, and they're going to have larger vacuoles than animal cells. Here's the things that they share in common: nucleus, membrane. They both have ribosomes. They both have mitochondria. This is really important. Plants also have mitochondria. They have to break down the sugars that they make. They both have cytoplasm as well. So here are a T-chart with it. Plant cells are box shaped. They have a cell wall, chloroplast, big vacuoles. They're green because of the chlorophyll. Animal cells are round, no cell wall, no chloroplast, small vacuoles. All right, let's go through this chart. You try it before I click through it. Do prokaryotes have a cell wall? Yes. Cell membrane? Yes. Membrane-bound organelles? No. All right, nucleus? No. An example of a prokaryote? Bacteria. You could say E. coli. Plant cells, cell wall? Yes. Cell membrane? Yes. Membrane-bound organelles? Yes. Nucleus? For sure. Example, how about a cactus? We're here in Oman. All right, animal cells, cell wall? Yes. Oh, excuse me, no. Woo. Brain fart after lunch. Animal cells do not have a cell wall. Cell membrane? Yes. Do they have membrane-bound organelles? Yes. Nucleus? Yes. Example could be a human cell. It could be an intestinal cell. Notice how they all have a cell membrane. The cell membrane is really important, and we're going to do a whole other video on that in a little bit. All right, organelles inside of eukaryotic cells. The structure of organelle contributes to its function. You can think about this with the mitochondria. It's set up to make energy. All right, the jobs of cells. There are three main jobs that we talked about in class: making energy, making proteins, and making more cells. We didn't get too much into this last one, so I won't really put it on your test. All right. Which organelles are used in capturing and storing energy? Go ahead and pause the video and think about that one. All right, here we go. Hopefully you got mitochondria and chloroplasts, also the cell membrane, because it had to allow food in. Lysosomes help to digest food. Vacuoles and vesicles hold it. The main ones I'm worried about for your class here, introductory biology, is the mitochondria and the chloroplast. Right? The mitochondria take in oxygen. They're going to use glucose, and they're going to oxidize it and make lots of ATP. Chloroplasts make that glucose. All right, here's a standard model of a cell from Khan Academy. This is good to know, but not all cells look like this, right? Your warm-up in class today was actually look at different cell types and note how the structure of an actual cell might lead to its function, like a muscle cell, what it looks like compared to, like, say, a red blood cell or an intestinal cell or a nerve cell. They look quite different, which is right here. Good. So here would be an example. The nerve cell, the structure of at least its function, is able to transmit messages very fast. You can see the red blood cell is concave. It's able to carry oxygen. And white blood cells are able to eat up invaders in our body and then send out messages to our lymph nodes to make antibodies as well. All right, let's go through the process of protein synthesis. You've done this on a molecular level of going from DNA to RNA in the process of transcription, and then from RNA to a protein in the process of translation. But now let's see it on a cellular level. And this is going to be called the endomembrane system. And it's an important thing to know for biology. Here's the nucleus. And inside the nucleus, we have DNA. DNA is going to be transcribed by RNA polymerase and enzyme to make RNA. RNA will exit the nucleus and go to the endoplasmic reticulum, on which are ribosomes. We call that the rough endoplasmic reticulum because it looks rough. It's got little bumps on it. This RNA can also migrate to free-floating ribosomes within the cell as well. For this instance, let's use the ribosomes on the ER. All right. The ribosome translates the mRNA message into a protein. Remember, tRNA brings the amino acids. The amino acids are joined together, and they form a protein. That protein then goes through the ER, and it is then leaves in a vesicle to the Golgi apparatus. And the Golgi apparatus looks like a lot of pita pockets put together. So we've made this protein. It's starting to take shape, etc. But we take it here, and this is where it's going to be modified, and then it's going to be shipped out of the cell. So here, the nucleus, it had the DNA. The nucleus is connected to the endoplasmic reticulum. The RNA makes its there. The protein's made, and then the protein goes in a vesicle to the Golgi apparatus. In the Golgi apparatus, this protein is modified. All right, it might have markers added to it. It might be put together to make a bigger protein, etc. But it's modified to do what it needs to do. Maybe this is human growth protein or insulin, etc. It then leaves the Golgi apparatus once it's done being modified, once it's all refined, and it is sent out in a vesicle to the blood to go do its job. And so now it's going to go off and do its work. So think about it kind of like you in high school, right? We're gearing up on graduation. It's in just about a week. And so we're sending the seniors off in these vesicles to go to school and do their stuff. All right, I encourage you to go to cellsalive.com to work on the organelles, right? And so you don't need to know all of them, but you should know the ones involved in protein synthesis, which would be the rough the Golgi apparatus, these vesicles, the nucleus, of course. You should know the nucleolus, which is this dark area right here within the nucleus. It's going to make ribosomes, which are important in protein synthesis. You should know about energy capture and storage. There's a mitochondria. If you click on the plant cell, you'll see the chloroplast. And then you can also learn about cellular division, how the centrioles are going to help to separate chromosomes. All right, so organelles that deal with energy, chloroplasts and mitochondria, making proteins like we just went over, nucleus, ribosomes on the Golgi apparatus leaving the cell, and then if it just goes from the nucleus to free floating ribosomes, those are going to be proteins that stay within the cell and that do intracellular work. So say that there's like little proteins here that are free floating in the cytosol, these red ones. So if the RNA goes to one of these, it'll make a protein that just works within the cell. The nucleolus makes ribosomes, and then making more cells involves the nucleus, right? DNA, copying it during the S phase, and then centrioles to pull it apart. We didn't get to that in this unit, so you can skip that. All right, the cell membrane will be the function of our next video. It's very important. It's going to be made of that phospholipid bilayer, which we talked a bit about in the chemistry of life. It's going to have a non-polar interior. The outside will be polar. It's going to be fluid, mosaic, etc. All right, it controls what enters and leaves the cell. Let's do a couple practices here now that you've gotten to pause the video and hopefully see what you know. So go ahead and pause it and see if you can figure these out. Here we go. Did you get it? Oops, I'm sorry. All right. If you thought I was pointing to the white ribosome, sorry about that, but it's pointing towards the cytoplasm. We have these lysosomes, which are going to help to recycle waste and break apart things. The nucleus holding the DNA. The ribosomes, sorry, I just had some visitors, making proteins. Again, here's the Golgi apparatus, EER, etc. All right, I'll try and be a little clearer with the labels there. I think they got messed up in PowerPoint. All right, same thing for a plant cell. All right, hopefully you got these. Let's see if there's any we haven't gone over. Nucleolus making the ribosomes. All right. Sorry, I had to say goodbye to some seniors right there. Okay, endosymbiosis. And so this is the very last lesson. We watched a TED Ed video on it. We did a little bit of guided notes and did a little bit of thinking about some of the evidence for it. So endosymbiosis, what is it? So endo means to enter in, and symbiosis means to live together, to work together. And so what Lynn Margulis, who is pictured here, proposed, and she was quite ridiculed for it, but she's absolutely right, is that eukaryotes evolved via endosymbiosis. So at some point, a cell engulfed an aerobic or engulfed the mitochondria, and then another one engulfed a chloroplast afterwards, and they're able to live together, right? And this led to eukaryotic cell development, right? The nucleus was formed by infolding in a membrane, but the mitochondria was endocytosed into the cell, and so was the chloroplast. Which one was endocytosed first? Well, hopefully you can see here is the mitochondria, right? Because all plants have mitochondria, but not all animal cells have chloroplasts. So therefore, the mitochondria was first, and then all these cells have mitochondria, and these ones also have chloroplasts. So both of these were endocytosed into the cell. Those are the two organelles that were brought in. What is some evidence for this? Well, some evidence that supports this is that they each have their own DNA. All right, and that DNA inside the mitochondria and the chloroplast is circular. It's very similar to bacterial DNA. None of the other organelles have their own DNA. The endoplasmic reticulum, the Golgi apparatus, they do not have their own DNA. They have their own ribosomes, which are similar to bacterial ribosomes, and they divide on their own. So if you remove all the mitochondria from a cell, the cell can't make more of them. The mitochondria kind of divide just like bacteria. They also have two membranes. They have their own membrane from when they were their own bacterium, and they have a membrane from when they were absorbed by the cell. And that was what we saw on the TED Ed video. And then I kind of went down a rabbit hole with with that on ChatGPT. It's pretty cool. Anyway, let's move on. Up here are my kids. They're reminding me that it's time for me to go exhibit some wellness and to go take care of them. I hope you found this video on cells to be helpful, and you keep crushing.